This video is on the second important topic in cultivation of bacteria, and that is culture media used in cultivation of bacteria. So let's start with a short introduction. Culture media is a source of nutrient for microorganisms. Now, as we all know, microorganisms are able to grow and multiply in nature very easily. They can grow in several environmental conditions. But when we talk about isolating and cultivating these microorganisms in our laboratory, we should provide these microorganisms with culture media, or we can say a nutrient media. Now these nutrients which we provide through a culture media, these nutrients are the source of energy for these microorganisms. The nutrients are important for microorganisms for synthesis of their cellular components and energy production. So a culture media should be formulated in such a way that they must be able to provide a balanced mixture of nutrients so that the microorganisms can grow on optimum levels. Now when we talk about studying this microorganism in our laboratories, then we have to first of all isolate and cultivate these microorganisms in a laboratory. Now each and every microorganism have different nutritional requirement, and on the basis of the nutritional requirements we have to design a culture media, or we have to formulate a culture media in such a way that they must provide a balanced mixture of nutrients. These nutrients are very important for microorganisms for synthesis of their cellular components, energy production and multiplication. So we should provide these microorganisms with appropriate nutrients, appropriate environmental conditions, so they can grow on the optimum levels. Now, as we all know, there is a huge variety of microorganisms in nature and the nutritional requirement also vary. So a single media cannot support the growth of all microorganisms. Now, as I told you before, there is a huge wide variety of microorganisms which are present in nature, and each and every microorganism have its own requirements. So a single media cannot fulfill requirements of all these microorganisms. So here a nutrient media should be designed in such a way that fulfills the nutritional requirement of that particular microorganisms. So here on the basis of the nutritional requirements, the culture media should be designed. Hence different types of media are designed for growth of different types of microorganisms. So as I told you, on the basis of type of microorganism, on the basis of nutritional requirement, here different types of media are designed. Now for isolation and cultivation of microorganisms, we use synthetic media. Synthetic media is nothing but a nutrient media. Now here when we are going to isolate and cultivate these microorganisms in our laboratory, the exact composition of the nutrient media which we are going to use, that should be known to us. And such nutrient media whose exact composition is known as a synthetic media. Synthetic media is a nutrient media which is made up of chemically defined nutrient. The exact quantity and quality of nutrient are known. As I told you before, synthetic media is the media whose exact composition is known. The quality and the quantity of the nutrients which we are going to add in this media are known. Now there are some common ingredients that are used in each and every media preparation. Now what are they? First one is peptone, second is meat extract, third one is yeast extract, fourth is sodium chloride, fifth is inorganic nutrients, sixth one is water, the most important ingredient, and seventh one is solidifying agent. Now let's see these ingredients in detail. Now let's see the common ingredients which are used in media preparation. First one is peptone. Peptone is a protein hydrolate that is prepared by partial proteolytic digestion of meat, soya and gelatin. Peptone is a source of carbon, energy and nitrogen. So here peptone is an important source of carbon, energy and nitrogen. The amount of carbon, energy and nitrogen which is important for synthesis of cellular components, growth and multiplication is provided by peptone. Next is meat or beef extract. Meat extract contains amino acids, peptides, nucleotides, vitamins, organic acids and minerals. So here you can see meat and beef extract is an excellent source of nutrient. It provides many substances. Meat extract provides vitamins and growth factors for microorganisms. Yeast extract. It is an aqueous extract of brewer's yeast. It provides amino acid, nucleotides, growth factors, minerals to microorganisms, as well as it is an excellent source of vitamins. Next is sodium chloride, that is NaCl. The normal osmotic pressure of a living cell is 0.85% of NaCl. So here in nutrient media the NaCl concentration is added in 0.5% of NaCl. Now what is the role of NaCl? NaCl maintains the osmotic balance. So the normal osmotic pressure which is present on the cell is 0.85%, and here the NaCl concentration which we add in the media is 0.5%, and the important role of sodium chloride is to maintain the osmotic balance of the cell. Now the fifth one is inorganic nutrients. Most of the inorganic nutrients are present in natural media, but in case of synthetic or specially formulated media we have to add this inorganic nutrients. Now inorganic nutrients such as iron, potassium, magnesium, manganese, phosphate, sulfate are added in the media. Now see, as we all know, microorganisms can easily grow and multiply in nature, and when they are present on the natural media, or when we can say when they use the sources which are present in nature as their energy source, or we can say source of nutrients, these sources provide inorganic nutrients. But in the case of cultivation of these microorganisms in laboratory, when we prepare a nutrient media for these microorganisms, we have to add the required amount of inorganic nutrients. Now inorganic nutrients such as iron, potassium, magnesium, manganese, phosphate, sulfate are added in the media. As per the requirement of the microorganism, the inorganic nutrient is added in the media. Now sixth one is water, the most essential component of the nutrient media, and water is essential component of a living cell. Water accounts 95% of dry weight of cell. Here we use water as a base, an important component in media preparation. As I told you before, water is an important component of the media, and the 95%, it accounts 95% dry weight of the cell. So it is the basic and the most important thing. Here we use distilled water in media preparation. Now why we use distilled water? Because distilled water is used because it has a fixed composition and it is free of impurities such as calcium and magnesium which are present in tap water, which can combine with media components and form insoluble precipitate. These precipitate are formed at high temperatures during sterilization. So here distilled water is used. Now here water is the important component and we use distilled water. Now why we use distilled water? Because the exact composition of distilled water is known, okay, and the tap water, it contains some impurities like calcium and magnesium. Now what these impurities can do, when we use tap water and add all the nutrients and when we go for sterilization, these impurities which are present in that tap water, that is calcium and magnesium, they can form insoluble precipitate because they can combine with the nutrients which we have added in the media, and they form insoluble precipitate at high temperature, and thus they can spoil the nutrient media. So here it is important to use distilled water. Now the seventh one is solidifying agent. Agar powder is used as a solidifying agent in nutrient media. Agar was extracted with hot water from various species of Red Sea algae like Giladium pacificum and Giladium japonicum. So here agar is a solidifying agent which is used in media preparation, as well as agar was extracted from a seaweed, that is a specific seaweed known as Red Sea algae like Giladium pacificum and Giladium japonicum. Now there are other solidifying agents are also present, but why we use agar? Because agar is an excellent solidifying agent first of all, and it melts at 98 degrees Celsius, that is during sterilization this agar melts down and it solidifies at 42 to 45 degrees Celsius, that is after sterilization when the temperature drops down to 42 to 45 degrees Celsius, the agar solidifies, that is this temperature is hand labeled. Agar forms a very clear transparent gel, so diffusion phenomena can be easily observed. Now gelatin is a proteinic substance. Now gelatin is another solidifying agent, okay. Gelatin is a proteinic substance prepared from bones. It is soluble at temperature of 40 degrees Celsius and gets solidifies at 28 to 30 degrees Celsius. So it has a disadvantage as compared to agar. So it is not used due to its limitations. Now gelatin is a proteinic substance which is prepared from bones, and the temperature at which this gelatin is soluble is 40 degrees Celsius and it gets solidifies at 28 to 30 degrees Celsius. Now this is the limitation we can say of gelatin. So here we use agar. Now let's see preparation of culture media, a basic idea for preparation of culture media. In preparation of culture media, some naturally occurring substances are used. For example, milk. Such substances should be sterilized before used. Here all nutrients are commercially available in powdered form. Now in some cases, cases of media preparation, we have to use naturally occurring substances like milk. Now such substances should be sterilized before they are used, and generally all the nutrients are available in powdered form. Now what are the steps in preparation of nutrient media? Appropriate volume of distilled water is taken. First of all, you have to decide what amount of media you are going to prepare, whether it is 100 ml or it is 1000 ml or whatever. So appropriate amount of distilled water should be measured by using a measuring cylinder, and you have to take this distilled water in a flask. All media components are weighed as required and dissolved in appropriate volume of distilled water. Now according to the volume of distilled water, the media components are weighed and then all these media components are dissolved in the distilled water. The pH of the media is determined and adjusted to optimum levels or appropriate levels. After adjusting pH, solidifying agent, that is agar, is added and is boiled to dissolve. Now once you add all the nutrient components, you have to measure the pH, and once you determine the pH, then you have to adjust this pH to the optimum level of that desired microorganisms which you are going to isolate. Now after adjusting the pH, further you have to add the solidifying agent, that is agar, and after adding agar you have to boil that media until the agar gets dissolved. Further, the media is sterilized by autoclaving or by filtration. Now sterilization is the most important step. Now you can sterilize the media either by autoclaving or by filtration. Now after autoclaving, the media is used for isolation and cultivation of microorganisms. Once the media is sterilized, it is cooled down, then you can further use this media for isolation of microorganisms and cultivation of these microorganisms. Now this was the basic idea of media preparation. If you liked my video, please like, share and subscribe to my channel, and thank you for watching.